This is a video about dihybrid crosses, so it's kind of the big boy squares. We've talked about a monohybrid cross, mono for one, dihybrid, di for two, and of course hybrid, you're familiar with that term. You've seen hybrid vehicles that are both electric and gas. Hybrid also refers to the fact that there's a cross, so it's kind of a duplication of terms. Dihybrid crosses. So for a dihybrid cross, sometimes there are traits that are linked or related to one another, and we consider both of those at the same time. When we talk about both crosses, things like seed color in peas and whether the seed is wrinkled or round, these are some of the things that Gregor Mendel looked at when he first discovered all the different aspects of genetics. Just a reminder, remember that Gregor Mendel gave us a lot of what we're doing today. If we call him the father of genetics. So considering two traits together means that we are looking not just at like big T, little t, or big T for tall, but we might be looking at big T, little t, and maybe talk about round seeds. Round is dominant in pea plants. So big R, little r, where the little r is the recessive that refers to wrinkled seeds. Remember, we always try to select in complete dominance or simple dominance, we select the letter of the dominant trait, and then the capital of that letter is the dominant, and the recessive is the same letter but lowercase. So let's look at a cross in pea plants that considers the traits round seed, that's dominant to wrinkled, and yellow peas, which is dominant to green, which is kind of puzzling because all the green peas I've ever eaten have been green and not yellow, but that's, of course, probably a selected, we select and produce the crops that are most pleasing to our eye, and I think green peas probably better looking than yellow. So that's where we are. Now, I'm going to do the X format on that first, and the X format you can see down at the bottom on your PowerPoint would be big R, big Y, little y. Same type of thing we did before, except now we've got two traits together. And of course, we are crossing, very common cross is a double heterozygous. In other words, something that's heterozygous for both traits. So we've got an identical parent, the other parent, and that's big R, big Y, little y. Remember, yellow is dominant, capital Y is that dominant yellow gene, and big R is round. How do we make the actual square? Well, it's not that much harder than what we did before. Matter of fact, that's what I recommend you start at, is make a box, just like you did before, maybe make it a little bigger. We're going to split it down the middle, and we're going to split it down the middle, okay? So down the middle and across the middle. That looks a lot like our monohybrid cross that we did. So the monohybrid cross was for one trait, remember, two alleles for one trait, and this dihybrid cross, we're going to need more squares because we've got more traits. So we're going to split this one, split that one, split this one, and split that one. So that's the easiest way to make a good dihybrid cross grid. And notice that we've got 4 by 4, which is 16 boxes. How do we divide up the alleles? Well, let's look at our X format here. We've got big R, big Y, little y, and we've got that for the top parent, and we also have big R, big Y, little y for the side parent. But there's an easy way to make sure we get all the possible alleles that can be passed on. Remember, this parent can either pass a big R or a little r, and a big Y or a little y. It can only pass one trait from each set of alleles. So, or can only pass one of the alleles from each trait, would be a better way to say it. So here's the pattern. Here's how you do it. First of the first, with first of the second. Okay? What does that mean? Well, that means big R, big Y, because we take the first allele of the first pair and the first allele of the second. Then we take the first of the first with the second allele of the second pair, which would be big R, little y. And then we switch to the second of the first pair, which is the little r. Second with the first would be little r, big Y. And then second with the second, so in other words, second of the first pair and second of the second pair gives us little r, little y. Try it on the side. See if you can set that up. As we go through this, you can see already both parents are identical, so both parents are going to be identical on the square. So again, first with first, the first of the first pair, and the first one of the second pair would be big R, big Y. The first of the first pair with the second, big R, little y. And then we take the second one in the first pair with the first one of the first pair. So we've got little r, big Y, and then little r, little y after that. All right, so how do we distribute these? Well, the easiest way is to just say we do what we did before. Whatever's here goes all the way across. Whatever is here goes all the way down. So let's see how we do that. Well, we're going to drop a big R and a big Y down, and we're going to take a big R and a big Y across. So I'm going to go ahead and take the big R and big Y all the way down. Big R, big Y, big R, big Y, big R, big Y. So that set of alleles is passed all the way down. This set of alleles all the way down. But we're going to try to leave space for the other R because in the end we put the R's together and the Y's. So we've got big R, little y, big R, little y, big R, little y, big R, little y, big R, little y. And then we take across the big R, big Y. We've got a big R and a big Y. Okay? So hopefully you can see that pattern. Now we've got a big R, little y, big R, little y. We've got a little r, big Y. Notice that I put the two R's together and the two Y's together, but we just carry everything across and carry everything down for each column, and we end up with all the possible crosses that would occur, and not only that could occur, but in the same percentages that they will occur. So this is how we could predict what offspring are going to look like for these two traits. So we've got big R, little r, big Y, and we're going to carry this again all the way across so that we've got this represented in that row, and we've got each header represented all the way down the column. So let's see. We're going to go ahead and I'm going to finish this up, and you could do the same on your paper. I think I've got a key at the end so you can see what it's going to look like. So we've got big Y, we've got big R, we've got big Y, little y, and going to finish this all the way across, and when we get through, you should see that this is what we end up with. And on the screen here, we've actually represented what the P's look like. So big R would be round, and big Y would be yellow. Now remember, just like our inheritance before, if there is a dominant allele, it doesn't matter what the other one is. The dominant one is dominant, and it shows up. So notice that wherever there's a big R, there's a round P. The only time you're going to get a wrinkled pea plant or pea seed would be when there's little r. It's wrinkled. Little r, wrinkled. Little r, wrinkled. And little r, wrinkled. And the only time we're going to have a wrinkled and green together is when we have little r, little y, because there's no dominant gene there, so the recessive shows up in both cases. Now, just like before, we've got a phenotypic ratio and a genotypic ratio. The genotypic ratio, the way you'd keep track of that is you would write like a scoreboard. You would write big R, big R, big Y, and then you'd write how many there were. So we'll start out with big R, big Y, and then you write a number. How many are there? Well, there's only one. One. And then we go to the next type. Big R, big Y, little y. Well, here's two of them here. It looks like those are the only two. So, again, big R, big Y, little y. There's two. And you go all the way down, and every time there's a different genotype, remember genotype means the actual type of genes that are there, the actual letters. Each time you find a different letter, you write it down and then go count how many there are. That's how you get the genotypic ratio. Phenotypic ratio is what they look like. Okay? So when we do the phenotypic ratio for this, we go through, and it looks like you're going to have a 9 to 3 to 3 to 1. Okay? So, what do we mean by 9? Well, round and yellow. Count how many round yellows there are. 1, 2, 3, 4, 5, 6, 7, 8, 9. It's easier here because there's a picture of them. But all you really have to do is look at how many of them have a big R and a big Y. That's it. If it has a big R, it's round. If it has a big Y, it's yellow. Doesn't matter other than that. So we've got 9 round yellows, and it looks like we're going to have 1, 2, 3 round green. Okay, how does it get to be round and green? Well, it has a big R, and it has 2 little y's, because that's the only time green shows up is when there's not a big Y. 1, 2, 3. What about the other possibilities? Well, what about wrinkled and yellow? 1, 2, 3. And then we have 1 that is wrinkled and green. 9 to 3 to 3 to 1 is our phenotypic ratio. That is the appearance, the ratio of the different appearances. And that's it. That's the only way that these can come out. Now, does that mean that there's 9 pea plants out in the field? Well, if we had 16 total pea plants, then 9 of them would be the round yellow. But we could do this on a bigger scale too. I mean, you could figure out 9 over 16. 9 out of 16 are going to be round and yellow. And if there were 1,000,000 plants, you could figure a percentage. So if you figure 9 divided by 16, get your calculator, then we get 9 divided by 16. That says that it's 0.5625, or about 56%. Remember, when you get a decimal, multiply by 100, you get your percent. So we could figure out of 1,000,000 plants, 56% of them, 56.25, are going to be round and yellow. So that's the power of these dihybrid crosses. Just like the monohybrid cross, they can be used for large-scale numbers like that too. So we're going to move on and give you, this is the instructions again to how you set it up. And we just went over that, but they're there for you to read. And then this is a little more evaluation of what's going on. You can see down at the bottom, we've got the actual genotypes, okay? And we've got the actual phenotypes, and it's 9, 3, 3, and 1 for the phenotypes. It tells you all about the different occurrences and what they look like, so that gives you a little better explanation. Now I want you to try one. And we'll have the key up at the front desk so you can check it. And you'll have one of these on your review that you could actually work on. And there will be a question on the test that will be a dihybrid cross. So if you don't understand how to do this, come in and get some help, and you could figure out. You can do this. Everybody can do it. You just have to decide you want to understand it. So here's the one you're going to try. Tall is dominant to short. We're familiar with that. We did that on the monohybrid cross in pea plants, and round seeds are dominant to wrinkles. So we just did that in the other one, and we're kind of mixing the tall and the seed appearance. So again, you're going to show a cross of two heterozygous plants. So first thing I want you to do is show the X format, and maybe check that out to make sure that you're starting the right way, because if you mess up on the X format, then your whole Punnett square is going to be messed up. So what you might want to do is see if you've got the right X format for the cross, and then draw your 16 square Punnett square and see if you could actually set it up, figure out the crosses, and then figure out at least the phenotypic ratio. If not, do the genotypic and phenotypic ratios at the end. So good luck. Try that out. See how it goes.